As probably everybody knows by now, there early on in COVID was a link established between obesity and severe outcomes. And this turned out to be independent of comorbidities such as hypertension and diabetes. I'm going to show you just a few of the slides just to review this. And then we'll get into the study that we did at Stanford to test our specific hypothesis about what might underlie this relationship. But as you can see in some of the earliest studies, this is from China, there is a linear association between BMI here, starting even down at 20, and severe disease in the early COVID cases. And here, when you look at another study in France, these are all small studies at the beginning, they also showed that the odds for mechanical ventilation was about 7-fold increased in individuals with a high BMI. Wasn't statistically significant for BMI over 30, but over 35, you could see this statistical significance. Again, this is a small study. Then in New York City, the next big wave, they showed the same thing. You can see that hospital admission was increased 2.5-fold for BMI over 40. And ICU admission was also 1.5-fold increased in those with BMI over 40. And then in Southern California, now we're getting into larger studies. This is now up to 7,000 people in the Kaiser healthcare system. We can see with high BMIs, the risk of death was increased for those under age 60 for BMI over 40, highly significant. For those over 60, it was only significant in the very highest BMI. And in this particular study, this wasn't borne out in all studies, but the risk appeared to be particularly high in males. And so then I'm not doing all the studies, but this meta-analysis of 75 studies on obesity and COVID-19 basically shows this following summary here, that the risk for testing positive is slightly increased. The risk for hospitalization is increased. ICU admission and mortality are all significantly increased in association with obesity. And I wanted to point out, since the early studies really focused on BMIs of higher than 35 or 40, that actually the association is linear and begins with a BMI of around 24. So these are various severe COVID-19 outcomes in a large all-payer database of over 3 million patients, where you can see hospitalization. These are all, the horizontal axis is all BMI. Hospitalization starts going up after a BMI of about 24, ICU admission, mechanical ventilation, and death all start to go up with BMIs above the average, normal body weight range. And this is also split up by age group. You can see that in every age group, you see the same association, that BMIs above 23, 24 are associated with increased risk. And I thought I would mention visceral obesity, which is a topic of great interest and for which there's very little data published at this point, at least not large studies. But there does appear to be an association with visceral obesity per se and severe COVID outcomes. So these are two separate studies. On the left, this study that was published in Diabetes Care by Battisti showed that admission to ICU versus non-ICU was associated with higher VAT, which is visceral, and lower SAT, which is subcutaneous, independent of age and sex, and that the ratio, we use the ratio too, it's just a way of adjusting for total obesity. So you look at the proportion of visceral to sub-Q. This was associated with age and sex and BMI adjusted risk of ICU admission. So the odds ratio for each 20% increase in the ratio is 1.25. This is just ROC curves showing that BMI, shown in blue, is not as great a predictor as waist circumference or waist hip ratio. Both of these are not really great measures of visceral obesity, but they're surrogate measures. And as you guys probably know, the steeper your curve, the better predictor. And then finally, there was a lot of interest in all the severe cardiac outcomes that were reported, a lot at the beginning of COVID. And and we know that there's fat around the heart. So this idea that if obesity is related to poor outcomes, is it possible that the fat around the heart is contributing, or obese people have fatty hearts? And actually, there's data showing, and the background to this is also that people who have more fat around their hearts have more cardiovascular events. That's also a separate known observation. But here you can see that in this study, admission to ICU or death, no admission to ICU or death versus admission to ICU or death. In this comparison, there was no association with BMI. In other words, BMI was similar in these two groups, but the fat volume around the heart was significantly greater in those with poor outcomes, suggesting that maybe the internal fat, visceral fat, epicardial fat is actually one of the most important mediators of severe outcome. And here you can see the same thing. This is a separate study showing that the epicardial fat volume was significantly greater in those with critical disease versus those with moderate disease. And this also importantly is one of the few studies that was done in a specifically younger age group. Okay, so SARS-CoV-2 is not the first virus that's been linked to severe outcomes in association with obesity. H1N1 is probably the best studied example, where the odds ratio for death for those with BMI over 40 was 2.8, and those with BMI over 45 was 4.2. The molecular basis for this was not established, but the link was clear. So in our study, we really wanted to get into the molecular basis. So why is obesity a risk factor? And there probably are many reasons, but people with obesity are more likely to have metabolic syndrome with hypertension, endothelial dysfunction, hypercoagulability, inflammation, and maybe latent CAD. Only about half of obese people have this whole kind of metabolic syndrome going on. So if this is truly the link, it would be important to distinguish whether the group that's having the severe outcomes is actually this insulin-resistant metabolic syndrome subgroup. And that has not clearly been established. If we have time at the end, we can talk about that. Another possibility could be poor mechanical ventilation due to a heavy chest wall, and another could be altered immune response, and there's data showing poor response to vaccines in obese individuals. But there also is this interesting observation that ACE2, the receptor for the spike protein, is expressed by human fat cells. And potentially this virus is binding ACE2 and getting into the fat. And the reason this was of interest is that these old studies in the gene, autopsy databases, GTEx, showed that not only is ACE2 expressed in the lung and the GI tract, it's highly expressed. This is mRNA in adipose tissue. This is subcutaneous and visceral adipose tissue. And adipose tissue is a target for other viruses. As you may know, adipose tissue is comprised of many different cell types, not just fat cells, but adipocytes have been shown to be susceptible to infection with H1N1 type A influenza and adenovirus, and then various other cell types within fat have been infected with other types of viruses. This is SARS-CoV-1, not 2. So we hypothesized that SARS-CoV-2 might infect cells in adipose tissue through ACE2 and cause an inflammatory response, which could potentiate the severe COVID we see clinically. And this includes that adipose tissue could serve as a viral reservoir for prolonged replication, higher viral loads, shedding and seeding of adjacent organs, right? If it gets into the adipose cells or other cells in the adipose tissue. And then the other part of this is that when the cells are infected by a virus, they would be expected to have an inflammatory response, right? I mean, any fat cells, we know there's inflammation in the fat. My lab studies fat and inflammation in fat and immunometabolism in fat cells in obese individuals and in people who have metabolic syndrome or insulin resistance have more inflammation in their fat compared to healthy controls. So it's possible that the virus getting into the fat would incite an inflammatory response, which then, the regional inflammation in this fat could cause damage to adjacent organs. Okay, so here's just a cartoon that I modified from another author, but here is what people think about. Here's the fat around the heart, and the fat around the heart has no geographical barrier. It's just touching, touching the arteries, the coronary arteries, and touching the myocardium. So anything going on in the fat could really affect both the vasculature and the myocardium. But if the virus is actually binding ACE2 and getting into these fat and replicating, causing inflammation, these cytokines can then incite the typical things, which, inflammation will tend to cause steatosis, lipotoxicity, and fibrosis. So this can lead to cardiac remodeling, diastolic dysfunction, heart failure, acute cardiac injury, and even fibrosis in some of these long COVID symptoms, which are, still not sorted out. Okay, so the specific questions were, does SARS-CoV-2 infect human adipose tissue? If so, which cell types are infected? Does infection in adipose cells or other resident cells in adipose tissue promote inflammation? And finally, is ACE2 expressed in these cells? And if not, is it coming in through some other entry portal? All right, so here's an overview of the study. So we were so fortunate to have our surgeons jump on this, and we already had established relationships with them. So we started this basically in, I think, April or May of 2020, and very quickly, but a lot of this work, which now we're, doing a lot more details. It's actually published in bioarchives. It's being re-reviewed in Science and Translational Medicine because we're doing some fancier tests. So it's not in the peer-reviewed journal yet, but you could find it on bioarchives. So we harvested the tissue from both bariatric surgery patients. We got SAT, which is subcutaneous, and visceral, which is that here. And then we also got cardiothoracic surgery patients so that we could get epicardial fat. And we also harvested pericardial fat. So here's the tissue. We have to do something called collagenase digestion to isolate individual cells so that we can do infections with the virus. So when you break down the collagenase, the mature adipocytes float to the top, and then the smaller cells come down in this pellet of stromal vascular cells that contains a variety of all the other cell types. So included in here are pre-adipocytes, and due to some of the difficulties in working with mature adipocytes because they float, some of the work we did was with pre-adipocytes from here that we can differentiate in culture and then infect with cells. So we differentiated the pre-adipocytes, and then we infected them with SARS-CoV-2 versus a mock condition on these days. Usually it takes 14 days to differentiate the cells. And then we also took these, the cells that were just separated, the mature and the stromal vascular. We took mature adipocytes that had floated to the top, infected those with SARS-CoV-2 versus mock, and then we took the total SVC with SARS-CoV-2 versus mock infection. So we have three different groups: the differentiated adipocytes, the mature adipocytes that floated to the top, and then the SVC, which contains the immune cells and other cells. So then we did real-time PCR for virus and protein and gene. We looked for some select typical inflammatory cytokines that we see in fats, so IL-6 and IL-8. We also looked at cytokines in the supernatant, so that's the secretome, to see what was secreted from the cells. We performed flow cytometry to look at which cells actually were showing evidence of viral infection within the SVC. And then to further explore that, we did single cell RNA sequencing to look at the virus and exactly which cells within the SVC are getting infected. And we can look at which pathways, this is all RNA, but which pathways are getting activated in those cells that got infected versus didn't. So what do we find? So here you can see, this is just the SARS-CoV-2 genome copy numbers. And we have three different tissue types here in a cardiothoracic surgery patient. So these are mature adipocytes and these are stromal vascular cells. The black bar are the cells that were exposed to SARS-CoV-2, and the white bar, which you can't even see, are the mock infected cells. So basically what this is showing is that in both mature adipocytes and stromal vascular cells from the subcutaneous adipose tissue, there's a strong signal of SARS-CoV-2 in the cells as compared to the mock condition, which you just can't see it because there was no virus detected. So this suggests that indeed these cells are being infected by SARS-CoV-2. We saw the same thing in epicardial adipose tissue, a strong signal in the mature adipocytes as well as the stromal vascular cells, but nothing in the mock. And then in the pericardial adipose tissue, again, we see the same thing. In the gastric bypass patient who is different from the cardiothoracic, not only in the depots harvested, but also that this patient is extremely obese. The BMI, well, I don't have the BMI of this one, but most of the cardiothoracic surgery patients don't have an extremely high BMI. But this patient is extremely obese but has no other COVID-19 risk factors. And in the subcutaneous adipose tissue, we see the same thing, virus infection of the mature adipocytes and the SVC, but nothing in the mock condition. And we see the same thing in the visceral. So this looks great. You know, this is what we first saw, but then we really wanted to confirm the findings. You know, maybe the virus is just stuck on the top of the cell. Is it really in the cell? Is it replicating? And what's it doing? So that's what we're still doing. But what we did show here is that, this is just a summary. We did more than one patient. We did 6 patients. So these are the stromal vascular cells from 6 patients in the subcutaneous and the visceral adipose tissue, and they all showed the same thing I showed you on the prior slide, which was single patients, but basically infection in the SARS-CoV-2 exposed cells, but not the mock. So then what we did to see if the virus is really viable in the cells is we did a remdesivir experiment where here are cells over 100 hours treated with SARS-CoV-2 and vehicle versus remdesivir. And you can see that those in the setting of remdesivir, it decreased replication of the virus. And this is in the subcutaneous adipose tissue, and this is in the visceral adipose tissue. So this does imply that the virus is in the cells and replicating because we can reduce it by remdesivir. The mature fat cells were the trickiest ones. And the only way we could really do this with remdesivir is to look at the differentiating ones in culture. So we were able to do that. So we don't see infection in the pre-adipocytes. I mean, again and again. So there's no infection. They don't get infected until they start to differentiate. So on day 3, day 6, and day 13, we can see signal of virus. And then when you expose these or culture them in the presence of remdesivir, which is the squares, there's decreased replication of the virus, again, implying that SARS-CoV-2 is not just stuck on the surface here, it's actually in the cells and replicating inside the adipocytes. And so then the question we had, you know, when you look, who's not doing that? When you look at RNA, you know, you have different primers. So we've been playing around with ACE2 primers to see whether we saw ACE2. We really didn't see a lot of ACE2 signal at all on any cells at the beginning. But we've been looking at these different isoforms. So we looked at full length and total ACE2 expression mRNA in adipocytes. And you can see that these are control cells. This is brand new, this is not even published. So, but in the differentiating adipocytes, you can see full length and total ACE2 are expressed at day 3, day 6, and day 13. So it does appear that functional ACE2 receptor is expressed in human adipocytes. We have not completed the Western blot confirmation of protein signal. So I'm telling you this with somewhat, you know, tentative conclusion, but it does appear that it's expressed in the adipocytes. And I'll get to the SBC in a bit. So now I'm moving on to the SBC. We wanted to know so we saw that the SBC was infected and the virus would replicate, but which cells within the SBC are they, right? There are many different cells. So we did some gating to look via flow cytometry at different subsets. This is just our gating strategy. We looked at macrophages. It's not super fancy, it's very basic. Dendritic cells, T cells, B cells, endothelial cells, and pre-adipocytes. And here's what we found. This is just the distribution of live cells. You can see that the majority were actually pre-adipocytes. Here are T cells, and macrophages were not the majority of cells. But when we looked at the frequency of a SARS-CoV-2 N protein on the cells, the only group that showed this was the macrophages. So, and here's the signal intensity again, macrophages above baseline. So these, the white bars are mock, and the black bars are the infected, infected meaning exposed to SARS-CoV-2 in culture cells. And this clearly shows that the only subgroup of cells within the SVC that got infected were the macrophages. This was, sorry, that's subcutaneous adipose tissue, and this is showing visceral adipose tissue. Again, this is the distribution of cell types, macrophages, T cells, pre-adipocytes. And again, the infection in the black bar, the SARS-CoV-2 exposed versus mock, you can see the signal is really limited to the macrophages. And here is the signal intensity. Okay, so then we thought, well, is it inciting inflammation in these cells? It's infecting macrophages, and is it inciting inflammation? So this is just our first look at this. So we measured via PCR in the cells, in the SVC, IL-6 and IL-8, and we looked at mature adipocytes and we looked at the SVC. We saw that in the SVC and epicardial adipose tissue, pericardial adipose tissue, and subcutaneous adipose tissue, and visceral adipose tissue, that the IL-8 and IL-6 were more highly expressed in the cells that were exposed to SARS-CoV-2 versus mock, implying that it does indeed, in SVC, incite inflammation. In the mature adipocytes, we did not see an increase in IL-6 or IL-8, except in the visceral adipose tissue. And visceral adipose tissue is generally more inflamed than subcutaneous adipose tissue. So for what it's worth, for these two genes, it looks pretty consistent that inflammation was increased in the SVC and maybe or less so in the mature adipocytes. So then we did Luminex evaluation of secreted proteins. So in these cell cultures, we collect the supernatant, and then we can measure all kinds of cytokines that may have been secreted by these immune cells and any cells within the SVC. These could have been secreted by the macrophages that got infected, but they also could have been secreted by other cells that somehow changed. For example, the pre-adipocytes, I don't have this data in here, but the pre-adipocytes actually changed in response to exposure to SARS-CoV-2. They adopted a pro-inflammatory phenotype, even though the virus didn't get into those cells. So I don't know where these cytokines are coming from, but it's very likely from the macrophages or the pre-adipocytes. And here you can see in these volcano plots that in the SVC from STAT and VAT, we get overexpression of numerous genes related to immune response and inflammation. So we see IP10, which is a cytokine that's already previously been associated with COVID storm, cytokine storm, and others related to hypoxia, angiogenesis, tissue remodeling, wound repair, and fibrosis. Basically, all the stuff you see in fat that's hypoxic or damaged and undergoing a response to that. Oh, sorry, these I don't know if I want to go through this, but this is just basically showing the known functions of these particular cytokines that are highly differentially expressed or differentially detected. VEGF, I just highlighted a few of them. All right, so I wanted to move on now to the RNA sequencing. So the single-cell RNA sequencing showed all of these different subsets of cells. You can see multiple clusters of cell types. They're all listed here. There are 22 clusters. But what we were really interested in is we were able to look at which of these clusters expressed SARS-CoV-2. And you can see on the right side very clearly that only one cluster expressed SARS-CoV-2. So we have these two clusters. These are macrophage clusters. We have C12 macrophages and C2 macrophages. So basically, a subgroup of the macrophages is the only group of cells that took up SARS-CoV-2, and it was the C2 cluster. This is a cluster enriched in IL-1 beta and CCL3, so it's a pro-inflammatory, a baseline subset of macrophages. C12 down here, on the other hand, is enriched in LYZ, not a pro-inflammatory type of macrophage. So even prior to infection, these macrophages were pro-inflammatory at baseline. And again, we just double checked this. We looked at CD68 on, this is the C2 and the C12. Both groups are macrophages, so they express the typical marker for macrophages, which is CD68, so that's expected. But again, we looked for ACE2. We did not see ACE2 transcript in either group, which was the same thing. I didn't show this, but on flow cytometry, which detects a protein signal, we also didn't see any ACE2 in the macrophages. So our original hypothesis about ACE2 seems to not be true for macrophages and to possibly be true for adipocytes. But we definitely did not see ACE2 protein or gene expression in these macrophages. So we looked at, what I want to show here are, we were just looking at maybe other entry portals in these macrophages. So remember, the C2 is the one that got infected and the C12 didn't. So this slide is comparing C2 to C12 in the mock condition. So this is before infection or in the group that didn't get exposed to the SARS-CoV-2. How do they differ? Why did the C2 get infected and the C12 didn't? So what we found, here's the ACE2, which again, you see no signal, but NRP, neurolipin 1 and 2, were both upregulated in the C2 cluster as compared to the C12 cluster. And cathepsin, CTSB, and CTSL were also upregulated in the C2 cluster versus the C12. And this CTSL has been shown by another group to actually represent a viable portal of entry, and we knocked it down. There was fewer, less uptake of SARS-CoV-2 in human and mouse cells. So these are of interest since they may represent other portals of entry into at least the fat immune cells.
A couple of minutes, Tracey, a couple minutes.
Okay, thanks. I know we're running behind already and I'm not helping. So, so these are differentially expressed genes and associated pathways enriched in infected versus bystander. So then what we're doing here is we're looking only at the C2 macrophages, so only the ones that took up SARS-CoV-2. And we're looking at within that cluster, about 85% of the cells took up the virus and 15% didn't. So the red bar here is SARS positive and the gray bar is SARS negative. So these are the cells that actually took up the virus that didn't. And what did they do? These cells, as you can see in this heat map, have higher red and yellow and red and orange are upregulation of genes. And these are the genes that were upregulated. They're basically pro-inflammatory genes, CCL4, MMP1, CCL8. Here you can see the pathways, the reactome pathways, again, basically related to immune response and inflammation in the cells that actually were infected. And then finally, I'm almost done. We were fortunate enough to get autopsies from 2 European countries. We had Germany and Switzerland autopsies. We couldn't get any from here. And these are just the clinical characteristics. They're basically mostly older patients, mostly non-obese with multiple risk factors. And in all of these, we showed via PCR a signal detection for SARS-CoV-2 in multiple depots of fat. So we saw it in periduodenal, perirenal, epicardial, subcutaneous fat clearly. And these are patients, this is important because the other work I presented is all in culture, right? So these are live patients who died from COVID who also had a PCR signal of SARS-CoV-2 in their fat. And here is a slide from Stanford where we were able to do in situ hybridization, looking for the SARS-CoV-2. Here it is, it's red, the immune cells are purple. And this is basically, the pathologists tell me that this is not in between the cells. These SARS-CoV-2 red, here you can see it here and here. These are in the cytoplasm of the fat cells, not outside. It just gets pushed to the edge by the lipid droplet. And you can also see here this infiltration of immune cells within the fat adjacent to these infected cells. And here's a zoomed out view of the immune infiltrate. And the red, again, is the SARS-CoV-2. And this is epicardial fat. So in conclusion, SARS-CoV-2 directly infects human adipocytes and macrophages. It replicates within adipocytes and macrophages and can be inhibited by remdesivir. The inflammatory response is initiated in SVC in multiple depots and elicits secretion of cytokines associated with severe COVID-19. ACE2 transcript and protein is not expressed in macrophages, but ACE2 transcript is expressed in adipocytes. And some other potential entry portals are overexpressed in the macrophage group that was susceptible to infection. And finally, autopsy studies in vivo demonstrate infection and evidence of SARS-CoV-2 in fat along with an inflammatory response. Thus, SARS-CoV-2 infects human adipose tissue where it can replicate and elicit an inflammatory response as we hypothesized, potentially linking obesity and or visceral obesity to severe COVID-19. And I'd love to give a huge thank you to my collaborators here. These are Catherine Blish, oversaw all the in vitro work in the BSL-3 lab at Stanford. These are the postdocs who did the work. Henry Chen in my lab did all the fancy adipocyte work and collagenase digestions. And this is my recruiter, our surgeons. And these are our wonderful pathologists who did the autopsy studies. Thank you so much for your attention.
All right, Tracey, thank you so much. You know, I've always felt there were reservoirs of virus somewhere, and I think we don't know all the reservoir tissues where it might be hiding, but this sounds like a good one. And
yeah, everyone's looking for you know, they're looking in other tissues as well. Yeah, I mean, we really continue to look at it. We're doing plaque tests and some fancier things right now to really be sure, but I think it's going to look like a reservoir. Whether or not it leads to long COVID is unclear, but it does appear to get into the fact.
So one thing I didn't know, so you were showing levels of RNA relative to mock. Of course, it's high, but what is the level relative to a traditional cell that we think of as vigorously replicating virus like lung epithelial? Are they equal, or are they hundreds of fold less than a lung?
I can address that. I didn't put in a bar graph for the autopsies, but the autopsy data is the best way to look at that. So what we looked at was the cycle threshold for fat compared to the adjacent organ. And so like for lung is always the highest, right? So lung would be very high. And then we looked at kidney and myocardium. And interestingly, you know, the cycle thresholds for the SARS-CoV-2 were a little on the high, you know, like the R range went up to, I think positive was up to 34, 36 or something. So they'd be around the high 20s and low 30s. And the lung would be higher, but in the fat around the heart, there was a higher signal in the fat as compared to the myocardium. So clearly, like kidney and lung would have cycle thresholds, I don't know, 20 or something, and this would be more like high 20s or 30. But it was there. It's not certainly not as high as the primary target organs, but you know, reasonable. You know, definitely respectable levels.
Right. Well, and as you said, people are finding other or thinking about other receptors than ACE2 anyway. So
yeah, there
may well be ways into those cells even without ACE2.
Yeah, and the macrophages can also just phagocytose the virus, right? So that's the other thing. Are they just phagocytosing the virus and it's in there and then it can replicate, you know, or you know, so that's also a possibility of how it gets into the mirror.
Right. By the way, the other trick that is helpful is to use primers for the virus that only detect the jumped RNAs, the subgenomic RNAs. So those get around the problem of loose virus or non-replicating virus.
Yeah, we actually did those. I didn't show them because the subgenomic method has, according to Dr. Blish, has been criticized, but we did do that and we showed the same thing.
Yeah.
And so that's why I think the best gold standard is the plaque assay will tell us if it's actually viable virus that can then affect another cell. And we actually do have that data, but I was told I can't present it because